Dear colleagues and distinguished guests, on behalf of my team, the organizing committee of Medical Student Journal Club, I welcome you to the 4th Pro et Contra Congress. How many of you have participated in the Congress last year? Raise your hands. Okay, the year before? Good. How many of you have been to the first edition of the Congress? Good. This year it will turn your life around. For the introductory part, it is my duty to say something smart and potentially funny for the audience. For the smart part of the speech, I've chosen to include determinism, of course, philosophical concept that is, by simple definition, the belief that all events are caused by things that happened before them and that people have no real ability to make choices or control what happens with or around them. As we're all made of nothing but atoms that are controlled by physical laws of nature, it all kind of makes sense, right? Apart from the horrifying idea of questioning free will, which is the super not funny part of my speech. Introducing determinism into medicine is, for example, to say a simple ankle sprain on a wet bathroom floor could be a cumulative result of an infinite number of actions that took place long before. The antique civilizations inventing bathrooms and so setting the stage for our scene. The carefree genes selected into our DNA that allow us not to pay attention to where we place our feet. And of course, the asteroid that made the replication of those genes even possible by erasing dinosaurs. And just to mention a few down the road from the Great Big Bang. At some point along this way, we, as medical personnel, interact with this chain of events, and together with the injured patient, we snowball down towards establishing diagnosis, prescribing treatment, and observing the outcomes. It really does put things into perspective, doesn't it? The idea behind Pro et Contra is also to put things into perspective. Sometimes only looking at some things from two different points of view can help us see the truth that is really underneath. Thank you. And before we continue with our program, it would be my honor to pass the word to the Dean of our faculty, Professor Dusan Šuput, who supported this project since its day one, and now still for the 4th year running.
Well, dear colleagues, I'm standing in between because I don't know what is the topic, so should I be pro or contra?
The topic is pro and contra. You should be pro.
Well, you mentioned determinism, and the funny thing is also, you also mentioned the atoms. And Laplace from Newton and so on really showed that determinism is the right thing. But if we look further, the principle of non-determined things that happen on lower levels, let's say on quantum levels, show that this is just the opposite. So it's the right thing to start, pro and contra. In medicine there are many things pro and contra, and I will tell you something that usually I tell when we have a ceremony. I'm giving diplomas, and I tell a story that was told by a dean at Harvard about 50, maybe even 60 years ago. And he said, well, the problem is that you have a diploma, but half of the things you learned are wrong. And of course they were a bit surprised. And then he continued, it's not a big problem, you know. The biggest problem is we don't know which part is wrong. This is also part of pro and contra. If you look at medicine, it changes. What I learned is maybe wrong now, but we are here to make things better. At that time they were right. They're the best choice we could offer the patients. Now choices are different. And the study of medicine is always pro and contra. Whatever you do, you interfere with normal state of a human being. And you have pro, curing disease, and contra, giving side effects. And the medicine is developing in a way that we hope to have a very small number of side effects, or very small side effects, and a big therapeutic potential. I think that just learning is not enough, and maybe it's even wrong if you study medicine. The right thing is to question, and that's why we are here. I wish you a very pleasant stay, very interesting discussions, and I'm really sorry that I cannot stay because I have a meeting at 10, and yeah, it's just the right time to go. Okay. Thank you.
So in Medical Student Journal Club, we are really proud and thankful today that we have two internationally recognized scientists here with us to share their knowledge and experience on a very recent topic. Probably most of you that are here from Ljubljana know about the very recognized article that was issued in New England Journal of Medicine just a few months ago. And today we have here with us the leader of the research team, Professor Tatjana Avšič Županc, to present their research and their first proven connection between Zika virus and microcephaly at the fetus. With her, we have here today gynecologist, her co-worker at the research, Dr. Vesna Fabjan Vodušek from Gynecology Clinic, Ljubljana University Medical Center. Please.
Good morning, dear distinguished guests, particularly students. Thank you very much for the organizers to give us the opportunity to tell us about our research and a little bit of introduction about the Zika virus and the infection that it causes. So first of all, I have to apologize our third co-worker out of the whole group. This is Jernej Mlakar, the pathology trainee who's supposed to be also presenting the case we had, who apologized this night of unexpected problems that he faced, so he couldn't attend here to be with us. So like it was already introduced, we will gonna present you our case, which was sort of a first proven association between the microcephaly and the Zika virus. As being microbiologist and virologist, and I always need to start with the virus if I talk about the viral infection. So Zika virus is a member of the flavivirus genus of the Flaviviridae family. And it's a typical envelope spherical virus that has single-stranded RNA genome. And this virus has some particular interesting part at the 3' end that has a secondary loop structure that form subgenomic flavivirus RNA. And this seems to be very important as it is essential for the pathogenicity as it inhibits host RIG1, which interfere with the interference and the innate immune system. So Zika, as I said, in phylogeny, being part of the huge group of the flaviviruses, belongs to the ones that are vectored by the mosquitoes, mainly by the Aedes mosquitoes. So how Zika virus is present to us in the nature? So Zika virus doesn't really need human beings in sylvatic cycle, that means in jungle. Zika virus, namely, is circulating between the primates, usually rhesus macaques, in the jungle, and the mosquitoes, and the virus is transmitted between the macaques and mosquitoes. But then somehow the virus with some sort of mosquitoes could come to the urban cycle, which means we at the time have outbreak. What means that infected mosquito infect uninfected human being. Human being at the time has the virus replicating in the blood. We call this viremia. And during the viremia, we have sort of a high concentration of the virus in our blood. And then us, human beings, are sort of a reservoir for uninfected mosquitoes. So that means when it comes to the urban cycle, human beings are reservoirs, and the virus is circulating between the vectors and human beings. And the main vectors are mosquitoes, and we believed until the epidemic that is currently going on in South America, that Aedes aegypti was the main principal vector, but now we are not sure that anymore. So again, in another picture, this is going on in the nature for probably millions of years, and human beings were not important for that. But when we come to the urban cycle, with a bunch of different other mosquitoes, we have outbreak or huge epidemic. So what Zika virus can cause? Usually, like all other arbovirus or vector-transmitted infections, viral infections, we have mostly inapparent infections. So that means without any clinical signs and symptoms after infection. But if infection appears, which we think in Zika infection, it's 1 to 4 infected people, it will show like a very mild febrile illness with fever, conjunctivitis is very prominent, joint pain, and very prominent are also maculopapular rash. So this is sort of a self-limited disease that usually lasts for a week with 2 days fever, and that's it. But we are facing lately, particularly after the outbreak or epidemic in French Polynesia, that Zika virus can also cause some more severe disease, like microcephaly in fetuses, or some autoimmune diseases like post-infection Guillain-Barré syndrome, and lately even we have been heard about the ADEM, which is acute disseminated encephalomyelopathy. So these are new entities that are autoimmune diseases caused like post-infectious diseases after the Zika infection. And this is something that is really worried and curious for us. So look a little bit at the history of Zika virus. It is a well-recognized virus from early 50s, from the past century. But nobody actually was really interested in that virus because it was all the time mainly present in the sub-Saharan Africa and then Southeast Asia, causing, just several human diseases, and nobody was really interested into that virus until it was an epidemic, actually an outbreak of the disease in Yap Island in Micronesia in 2007. So at the time, there was like 3/4 of the inhabitants of this Yap Island in Micronesia infected with the virus. So at that time, even myself, I started to be interested in the virus and also all the other infectious disease society. Then the next outbreak, actually, which we already can call epidemic, was caused in French Polynesia Islands, already involved a couple of thousand of people. And already there, we have been noticed some Guillain-Barré post-infection syndrome. And then the virus seems to follow that way through the Pacific and enter into South America, where we are now facing for almost 1 year the huge epidemic of the disease. So this is again how the virus was passing the continents of the globe of world. So how the virus is transmitted? The virus has been first found in rhesus macaques in in the forest of Zika in Uganda. Coincidentally, actually, the researchers at 1947 have been searching for yellow fever virus, which is also flavivirus, and coincidentally isolated Zika virus and called it Zika because it was in Zika forest. And then they found it also in mosquitoes. And they recognized that the virus can be transmitted and is transmitted with the mosquitoes. So they inoculated mice, baby mice, adult mice, and they found that the virus can be replicated in mice at the time. That was in early 1950, last century. But then they recognized that the virus can be transmitted with the mosquito bite to humans. And then, like I said before, from human to human, causing this epidemic urban cycle of the circulation of the virus. But what has been also found is been found that the virus can be quite in a high level, high viral load, also in saliva, not only in the blood, in urine, like in excreta, and also in the semen. So these are now new ways, new understanding how the virus also can be transmitted. So that means also sexual route, which is probably not that efficient as with the mosquito, and definitely with the transfusion. And the most important thing is that the virus probably could be transmitted from infected mother to the fetus. But that's are just new information from the last epidemic that started last year in South America. So why we have been very much curious about what is going on in South America. The most affected country in South America still now is Brazil. It is the most populated, and it's not the whole country that is very heavily populated. Northeastern part of the country is the most heavily populated and also has a perfect micro and microclimate for the mosquitoes to be long-lasting whole the year, 12 months per year. So we have at the same time very perfect level of having mosquitoes all the time and heavily population at the same time at the same place. So this is perfect for the urban cycle of circulating of the virus and causing epidemic as we are facing now. So Brazilians found the virus last year, starting somewhere March and having peaked in May, June. And then in early autumn, like October, middle October, late October, they have started to post some notes that in the part of northeastern part of Brazil, they're facing and counting increased number of microcephaly in those places of the country. And they have started to think that probably there is some connection between the infection with the Zika virus and the microcephaly. But there were doubts. CDC, WHO, nobody wanted to accept this information immediately from the Brazilian health organization because we have to be aware. Brazil and all South American countries are also endemic for dengue. You probably know what dengue is, another flavivirus that is the most spread flavivirus transmitted by mosquitoes all around the tropical, subtropical level of the globe. So dengue is present in South America for at least last century. As well as Brazil and South American countries were facing with chikungunya, which is not flavivirus, is a toga virus. But lately, the last 3 years, chikungunya entered into the South American and the Caribbean countries. And what is the problem? The problem is that all three viruses are causing very similar clinical presentation in people that are presented at all with the disease. So all started with flu-like syndrome, with the fever, muscle bone pains, with sort of rash, conjunctivitis. So it's very difficult to distinguish between the three viral infection based only on the clinical presentation. Even worse, it is by using serology or simple virological diagnostic, is also difficult to distinguish between the dengue and Zika virus, because these are both flaviviruses that very highly cross-react. So only with the direct detection of virus within the blood or urine sample is the one that can distinguish easily. So the problem started, as I said, with having at the same time all three viruses circulating, high level of Zika virus infection just enter into the naive people in country, and at the same time high increase in microcephaly reporting. So this was a very alert. And that's why Margaret Chan, who is the director of World Health Organization, posted in 1st of February this year an alert of the global awareness that Zika represent really a global alert. And they posted also a case definition for Zika virus, which is related to fetal microcephaly. So they said that fetal microcephaly is the one that should be molecularly or epidemiologically linked to Zika virus, with the absence of other conditions that are known to cause microcephaly. So that means that the pregnant woman is a confirmed case of Zika virus, or that the pregnant woman had sexual contact with the confirmed Zika virus case, or that there is a presence of Zika virus in amniotic fluid of a pregnant woman, identified by any molecular methods, or that presence of Zika virus is in the fetal brain tissue, again identified with the virological methods. Then, at the same time, it should be absence of other conditions that are known to cause microcephaly. We know that there are many conditions, environmental, genetic, and so far, so on. And also some other infections agents that can cause, for us, much more known until Zika, to cause microcephaly. So that means other congenital infection, exposure to toxic drugs, genetic abnormalities, and malnutrition in the placenta. So here we come to our case report, and I'm just giving now word to my colleague, Vesna Vodušek, who will continue, and then I will continue again.
Good morning to everyone. So here our case starts. Exactly, it started when the patient was presented to our department in October last year. So at that point, we didn't know what we are facing, because she came to us with a very severe problem. Her baby was sick. She already passed many exams, and nobody really knew what to expect, what was the cause of the problem of the fetus. So we were, as a tertiary center, the last part of this story. And to go through, she was admitted to our department in October. She's 25, now 26 years old, and she's perfectly healthy to this point of our visit. And the only thing that she mentioned as she came to us was that she lived and worked for quite a long time in Brazil, in Natal. So it's a northern part of Brazil, in the regions where Zika virus is circulating, and where the infection is possible. And she had some severe problems with high fever, severe poliomyositis, and a macropapillary rash at the beginning of the pregnancy, so in the first trimester, that was in February last year. She was not tested or any diagnosis was done because it's really expensive to do this in Brazil, and if you don't have the money, it's usually not done. But because Zika is supposed to be a very mild and unproblematic disease, they didn't do anything. They just prescribed her paracetamol, and the problems disappeared about a week later. Then she had regular visits at the perinatal clinic at Brazil. Several ultrasounds were made. The last was made in the 20th week of pregnancy, and after the papers, the development of the baby was perfectly fine, so they didn't find any anomalies at the fetus. Then later, during the summer, she moved back to Europe, and in the 29th week of pregnancy, another ultrasound was performed to see how the baby was growing, because the size of the uterus was a little bit too small. So that's one method how to look after a pregnant woman, to see if the baby is growing perfectly or not. And because of that, an ultrasound was made, and this ultrasound unfortunately showed us many fetal anomalies. So it was a symmetrical intrauterine growth restriction. So the fetus was small at whole. Especially small was the biparietal circumference and the head circumference. And again, the cerebral and the cerebellar structures were very, very pathological. So many examinations were done, especially ultrasounds, serology, to exclude any possible agents that could cause the microcephaly. And of course, also karyotyping was done. So an amniocentesis was performed in the 30th week of pregnancy, and the karyotyping was perfectly normal. So then she came to our department in the 32nd week of pregnancy, and of course we confirmed the intrauterine growth retardation. The weight of the baby was way below the 3rd percentile for its gestational age, and the placenta was very thick with a lot of calcinations, so it can be here nicely seen. So firstly, this is the head with the head circumference, it's the thalamus, and a lot of calcinations are already seen in the large brain, some ventriculomegaly, so it's the sign that it's seen when the brain is not developing rightly, and there's much more liquid in the central nervous system as usual, and also the placenta. It's very thick. It's usually between 3 to 4 centimeters thick, but in this case, it was in some cases also 6 centimeters thick, and there's several calcinations that can be seen. So we usually see such an ultrasound image in a viral infection, but we again performed all the serology to exclude any viral infection. And they were all negative at this point. But, so these are again some images from the ultrasound. So ventriculomegaly, here is very nicely seen that the brain is smaller than it should be, that the spaces between the brain and the bones are larger than it should be, so it's also the cerebellum and the brain is smaller, and again a thick placenta. And we have done also a magnetic imaging, an NMR. And here again, the same thing is seen. So the brain is smaller, some ventriculomegaly, and the gray matter of the brain is very, very thin, and the white matter is almost the normal thickness. So it's usually seen in very pathological development of the brain, and these babies usually are not capable to breathe, to see, to function by itself, so they are very, very sick. So because of all this, the parents were giving a poor prognosis for the neonatal health, and after a long discussion, the mother requested for medical termination of pregnancy, which was approved by the ethical commission at our clinic, and also by the Republic Commission for medical termination of pregnancy. So the procedure was performed, and she was delivered vaginally about a week later. And of course, after that, the autopsy was performed at the fetus, and also some additional genetic tests were done with the fetus tissue. So this is the picture of the poor baby. So it's really also growth retardation. The baby's weight should be around 2,400 grams at this gestational age, so it was a kilo lighter. And you can also see the upper part, so the head was very small, the cerebellar part is smaller, but the facial part of the head is normally developed. And at this point, we also didn't find any other developmental changes or pathological findings of the fetus. So, because the karyotyping was normal, we then performed some microarray analysis to the fetal tissue, and there were no genetic factors that could cause macrocephaly found at this microarray analysis. So we were facing the fact that something else caused, and of course the first thing that we thought about was still it was an infection. I can say at this point that when she told us that she had some fever-like illness that they called Zika, we also looked at the papers that wrote about Zika, and in October, the only thing that was said, it's a mild disease, don't worry about. 80% of the people even don't know that they have it, but we carefully have written everything down, and then at the pathological examinations, again, this feature came out, and it started all what happened later.
Please. Thank you very much. So the story goes on, and like my colleague mentioned, they very carefully listened to the patient, to the mother, and her description that she has sort of probably Zika infection in her third month of pregnancy, and like normally gynecologists and pathologists are not that following news about the viral infections around the world as we have to do, and we like to do, virologists. So it was very good that they marked this note, and then the trainee pathologist who is missing today, unfortunately, before he started to perform autopsy, he read that note, and he gave me a call. And he said, well, Professor Avšič, I remember that you have been lecturing us about the tropical viruses, flaviviruses. And I also remember you have mentioned chikungunya, which was for me very unusual name for the virus, but you didn't mention Zika virus. Do you know anything about Zika? I said, well, I know very much. And I didn't mention nothing about the Zika virus, particularly not to medical students, because I have to lecture in 1 hour 4 viral families, and because Zika was not really important for the medical science until this year. And he said, well, I have a fetus which I need to make to perform autopsy, and the lady was in Brazil, and probably there is some infection. Obstetricians are curious about that. Are you interested? Could you perform some diagnostic on that? I said, definitely yes. And at the time, I've already had, overnight some important reports that are between the network of us virologists about that connection that I showed you before, microcephaly increase in Brazil at the same places as they have Zika virus infection. So I'm already mentioning in my brain. So anyway, we have discussed and we have agreed which kind of material he has to take, particularly for virological examination, because this was, again, they have been very precise in their work. Also, pathologist was very precise, contacting me in advance, because for microbiology, particularly for virology or for RNA viruses, it is very important that you have very good fresh material. So the autopsy results actually showed, like we can see here, the Zika brain of our case and the brain of a girl who died for some other diagnosis, but her brain were normally growing at the same 32 weeks of gestation. So we can already see this difference in macroscopic examination. So microcephaly was around 1 percentile. Low placental fetal weight ratio was less than 3rd percentile. And like a colleague has already mentioned, the pathologists have also seen the calcification of the placenta after autopsy. And there was a gross microcephaly, micrencephaly, which means small brain, weighing only 84 grams, that is 4 standard deviation below the average. So when the sections of the autopsy has made, we can see from the frontal lobe further on that there were widely open sylvia fissures, small cerebellum, and the brainstem. There was complete, almost complete ageria, particularly in the frontal lobe, and internal hydrocephalus of lateral ventricle that have been seen in the sections. But then again, when we compare brain of the Zika patient and brain of the autopsy of a girl at the same gestational age that has no problems with the brain development, we can see very nicely complete ageria in the frontal lobe compared to the one of the healthy brain. Numerous calcifications, probably, I hope you can see them, frontal and the lateral lobe. And again, hydrocephalus due to damaged brain, particularly cortex part. So then the pathologists perform some hematoxylin eosin samples and also immunohistochemical labeling. And what we can see from A, B, C to D, great calcifications, particularly in cortical and subcortical part, and complete absence of gyration. Then also calcification with some filamental structures, which show some damaged axons and dendrites. The two down immunohistochemical labeling samples show the proliferate activated astrocytes over here that extend into subarachnoid space, and some activated microglia cells in the cortex could be seen. When they look at the spinal cord, the hypoplasia of the brainstem has been noticed, as well as severe Wallerian degradation of the lateral corticospinal tract has been seen. Nothing else, no other organ in histopathology, pathology has shown any abnormalities, only the brain. So they continue with the immunofluorescence staining on the cortical part of the brain by using a mother serum, because we have her serum, and what we can see here is a nice indirect immunofluorescence reaction, particularly in destroyed neurons, which can be confirmed that this is a neuron because there is a lipofuscin granules autofluorescence could be seen. This orange part, but this green part is immunofluorescence with the mother serum. So we use a bunch of negative controls to confirm that this is really specific. So, like I said before, placenta has this local calcification, and also there were no pathological changes in other fetal organs, nothing in umbilical cord, and nothing in the fetal membranes. That's all that has usually are checked with the autopsy of the fetus. And as colleague has already said, the medical geneticians has performed microarray to check the major possible causative, genetically causative agents of microcephaly. There are a bunch more ones, but they're very rarely shown. So then we continue in microbiology by looking at this brain tissue. And as you know, viruses can be only seen under the electron microscopy. We use transmission electron microscopy, and we were happy just having a new one. So we said, if there is a virus, we're going to see it. And definitely we have seen some vacuoles here, but there were not the virus with the vacuoles, but these ones. And we look at the high resolution, and we have seen some structures that could be viruses, and some disrupted endoplasmatic reticulum. And endoplasmatic reticulum of the cell is the place where the viruses are usually replicating, forming the new virions. And then we have performed from the frozen brain tissue some special slides that will look under the transmission electron microscopy, and where we have seen some structures being like between 45 to 55 nanometers in size that has this darker round shape and brighter interior. Which means that there is a viral application, and this has been shown already by some electron microscopy specialists in Center for Disease Control with some other flaviviruses. So we have been pretty sure that this is the virus, and in ER. So then we have performed negative staining after the ultracentrifugation of the brain tissue directly, where you can get only the virus structure if there is any virus, and we got it. And we have been pretty sure that there are viruses directly in the cortex of the brain, and that it seems that the virus is replicating. But we have to do further on. But the first thing, before we even do anything, we got like placenta, lungs, heart, skin, spleen, thymus, liver, kidney, and cerebral cortex. Agreed with the pathology trainee before autopsy. And we started to perform with the easiest method in virology that we are using now, with real-time PCR, directly checking for the Zika virus because we were suspecting, and we at the same time simultaneously look at the dengue virus and chikungunya virus. You remember I mentioned you that these 3 viruses are present in the Brazil, in South America, and Caribbean right now, causing epidemics and causing similar infection in human. And as we heard, the mother was infected in her third trimester of pregnancy, but at the time, nobody in Brazil has performed any virological diagnostics. So we were not sure if she was infected with Zika or dengue or chikungunya. And what we have received, and I was really shocked when we received the first result that morning, that only Zika virus PCR was positive, and when my colleague said me, yes, but only in the brain tissue, I said, I don't believe you. Please, could you confirm again everything? All 9 organs with all 3 PCRs. It shouldn't be only in the brain. Because I was expecting to have, the virus find in other organs, at least in placenta. They continue, they confirm, and they got the same result again. Only Zika virus PCR positive, only in the brain tissue. So I said, okay, I still don't trust, although we never had any Zika virus amplification before in our lab, so it couldn't be, any contamination like we used to say in microbiological lab. So then they have to use another gene to try to amplify. So first we amplify NS5 gene of the virus, and then I said, okay, look at the E protein and sequence the products. They sequenced the products, they said, okay, could you boss finally believe this is Zika virus? So anyway, we found also high level, that means high viral copies per milligram of tissue of the virus in the brain. So all the data that has been known at the time we got this, that was early November last year, we knew only how is the viral load in the viruses that you can cultivate in cell cultures, and in viremia, which was usually much, much lower, at least 3 to 5 copies, log copies lower than this. But it was similar what they have found in the semen. So we said something is going on. And then we continue, we immediately contact our colleagues in gynecology, colleagues in pathology, what we have discovered, and we said we have to work on this case further on. And as what we were doing in our laboratory, we tried to cultivate the virus. We are still doing in that process until now, trying to cultivate the virus in different cell cultures. But at the same time, we use the new generation sequencing, and we have been able to get the complete Zika virus genome sequence recovered from the brain tissue. And as I said, at the beginning, the case definition of WHO, so you have to rule out, when you discover something, you have also rule out all other causative agents, or environmental, or like they did, genetical causes of microcephaly. So we did on all 9 tissue samples, all these different PCRs, so like dengue, chikungunya, yellow fever, West Nile, tick-borne encephalitis, lymphocytic chorea meningitis, CMV, which is the most often teratogenic virus that goes through the placenta and infect the fetus. VZV, herpes simplex, enteroparvobenign, rubella, and toxo, which are also well-known teratogenic viruses that are causing infections and damage of the fetus. So all was negative in all 9 material of the fetus. Then we did a sequence analysis of our whole genome sequence compared to what has been known at the time in the gene bank. And we have found that the Zika virus that we detect in our case in the brain of our tissue from Natal, Brazil, actually is deriving from the French Polynesia. So it was another proof that the Brazilian strain of Zika virus actually came from this French Polynesia epidemic that caused epidemic 2 years ago. And we have found also some interesting amino acid changes at the NS1 and NS4B region, which probably after few months will show if these changes mean something important for the virus and the changes of the virus and so on. And again, what we had to do also in microbiology, like I said, and like my colleague said before, you know, we have to prove that there was really vertical transmission from the mother to the fetus. And we knew that the mother was infected with some virus, Zika, dengue, chikungunya. But at the time she came to gynecology department was already 6 months later, as she had this acute evidence of infection. So after 6 months, no PCR could perform in her blood sample. So we have to rely only on serology. And as I said before in my introduction slides, serology in flaviviruses is pain because all flaviviruses cross-reacting. So we had to perform all these tests, including all CMV, rubella, herpes, and blah, 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 that we really find by ELISA or by IFA, immunofluorescence, cross-reactive. So she has antibodies to Zika virus, IgG, and also to dengue virus, IgG, dengue 1, 2, 3, 4. She also has antibodies also to yellow fever virus. So how to prove now that she really had at the time Zika virus acute infection? So we had to use another very, very difficult test to use. This is neutralization test. That means we had to grow all those viruses, all those flaviviruses, and then use her serum and see how her serum neutralizes the viral infection in the cell culture. So we have been performing this for practically 2 weeks, and we finally found that actually she has the higher titer in neutralization test to Zika virus. So we actually prove that she really had at least the last infection with the Zika virus. We could not rule out from this that before Zika, because she was living there almost for 2 years, she didn't have also dengue, but at least the last one was Zika. So from this part, we can conclude that this was really the first and the strong evidence between Zika and microcephaly. So as I believe in that, and I convinced my colleague of that in December month, we have to write that down. We have to send it to the best medical journal in the world. If this is true, they will accept it. And if not true, and if we are not the first, so that was also some kind of race, but actually was not really race because nobody had that kind of results, still doesn't have that same kind of results yet, they will accept it immediately. And that was really the case, and it worked out fine for everybody. But our goal was just to help people, not to publish the best paper, because this was something that when I received and my colleagues received so many emails the night that the paper came out, was like, thank you, now I believe it. You know, people didn't believe that is going on. So this is association also of vertical transmission. So that means it's a teratogenic virus, another one, which severely affects central nervous system, damage placenta. It seems to be very neurotropic virus. We think that the possible location of the virus is in the neurons. We also think by our pathology department that probably the development arrested in the cortex of the embryonic age about 20 weeks in our case, because she was infected in 13 weeks of gestation. And we also think that the virus probably very, very slowly persists in the fetal brain. So I will give you the last words.
So to conclude our presentation, we are still at the beginning of the story because we have proven that Zika virus is a teratogenic factor, but there are more and more questions that are opened. Why, how, when, and so on. So we still don't know what to do in special cases, but at this time what we know is that a pregnant woman can be infected with Zika virus. So it can be done, or it's done with a mosquito bite, or she can also get infected with a sexual intercourse with an infected man. And we know that infected pregnant women that suffered the infection with Zika virus can pass the infection to the fetus. But what we still don't know is if a pregnant woman is exposed to the infection, how likely she will be infected. So if is there a possibility that the virus won't get a visible infection, or she will not suffer any major or minor effects of the infection. If the pregnant woman is infected, how the virus will infect her or her pregnancy? Will she be more, is there a more pathological pregnancy? Is she a high-risk pregnancy, like, for example, in flu, where she's more affectable to other factors, so she can also, the progression of the disease can be worse. She could have more a severe form of the disease, or even it can be also the opposite part. So less pregnant women can show these symptoms at the beginning or of the infection, and also how likely is it that the Zika virus will pass through the placenta to the fetus? So placentas can differ, and of course they are the first, I can say, wall that is protecting the fetus from everything, from the environmental factors, from the infections, from the bacteria, from the viruses, from everything what is going on in the body of the mother. And of course, we don't know what the ratio, what the likelihood is that the virus can pass the placenta and then go to the bloodstream of the fetus. We don't know if the fetus is infected, if the fetus will develop birth defect. We know that the immune system in utero by the fetus is very it's not very good. It reacts slowly. It usually even doesn't react to any pathological and any bacteria. So that's the reason why toxoplasmosis can cause so many defects, especially at the central nervous system at the fetus. So, but still, genetically, there can be a fetus that can react to this infection, and of course, in this case, he will not develop birth defects at the infection with the Zika virus. We even don't know if the pregnancy and the infection in the pregnancy may cause any harm to the fetus. So, is the brain the only thing that's infected, or there can be any pathological findings, and whether the baby will have birth defects, what I already mentioned. So will the pregnancy still go on. So a lot of discussion is going on also if the infection happens in the first trimester of the pregnancy. There can be a huge or a large group of women that are giving stillbirth and even a miscarriage to the pregnancy. So it's also possible that more and more pregnancy will get terminated by itself, by a natural way, because of the infection. And we don't know if a sexual transmission of Zika virus can differ the risk for birth defects. So it can be possible that if a pregnant woman is getting infected by the semen, that the progression of the disease will get milder or worse. So it's not clear yet what to expect. So on behalf of that all, CDC prepared some recommendations, especially for women that are trying to get pregnant or for women that are already pregnant, that especially for women in the reproductive age, that during the travel in areas where they can be infected with Zika virus, they should use a very good contraception. So they shouldn't try to get pregnant at that time. And if they are already pregnant, they should avoid to travel to these areas to diminish the possibility to get infected with Zika virus. And if the travel can be not postponed, they should use a good protection against mosquito bite, especially long clothes, mosquito nets, and so on. And they should use a condom during the whole pregnancy if there's any suspicion that her partner or her husband could be infected with the Zika virus in the last year. Because we know that Zika can be present in the semen for a very long time. It's not clear yet how long. They know it can be longer than 2 months. So that's also the reason why we shouldn't accept any sperm donation if there is any suspicion of an infection with Zika virus at the male. So these are the major recommendations that are done, and they're for this moment the only thing that we have and can deal with, especially when parents are coming to our department to ask what to do, how to do, and what could they expect. At this point, I will thank you, all the co-workers that have been worked on this project. So you can see it's a quite large number, and there's still a larger number that cannot be written here and anywhere. And of course, Professor Avšič, that it's still chasing the Zika virus. So it's a kind of her passion. And please, for any questions.
So again, on the behalf of the Medical Student Journal Club Organizing Committee, I would really like to thank you for taking your time and for being here today with us and sharing your knowledge and experience. And I have to say that we should be really proud that we have this opportunity, that we live in this moment, that we can learn from such great scientists from our own faculty, and we should be able to carry on this curiosity to our future careers. Thank you very much. And now for the students. For the students, I'd like to invite you to cooperate in our so-called survey, or actually the best debate competition of both of the days of the Congress. During the morning, you will receive to your email a link to the survey where you can vote for the best debate of today. And tomorrow you'll get another survey for tomorrow's best debate. You can only vote once, so think well before clicking. And the best debates, of course, will be awarded by the organizing team. Now we have a 10 minutes break where you can have some juice, cookies, and so on. And we'll continue at 11 sharp with the session rheumatology.